Thank you so much for joining us. We have a special guest today, Dr. Aaron Carrithers. Aaron is Chief Medical Officer of a company called Sequela, and he is an MD. He is a physician entrepreneur who helped start and run a couple of companies. He has all his degrees from Kentucky universities, from undergrad and from Transylvania, his MD from University of Kentucky. He did his postdoc in 2020, and he has undergone a lot of training at NIH. And today he will share with us his experiences in SBIR, STTR Phase I, in early stage development of medical devices and therapeutics, what you need to think about regulatory pathways as you are trying to figure out your way through their SBIR Phase I, thinking what will need to happen after that, and also what people need to think about in general at early stages of your companies. So on this, Aaron, please go ahead and share your screen. Give you permission to that. Yep, you have permission to do this. Please share your screen.
All right. Let me know if it's working. How about it? Can you see my PowerPoint?
Yes, we see your PowerPoint.
Okay. All right, so thank you, Eugene, for the introduction, and I just want to thank Eugene and Accelerate Health for having me today. Like I said, my name is Aaron Carrithers, and I currently serve as Chief Medical Officer and Director of Clinical Operations at Sequela Biotech here in Louisville, Kentucky. I also work with a consultant team where we help SBIR, STTR Phase I and II applicants further develop their commercialization efforts and delineate strategies for their product's regulatory clinical indication for use. With that, I'd like to transition into my talk and discuss how Phase I SBIR applicants can start to think about regulatory pathways early in their proof of concept, feasibility, and R&D studies. First, just as a disclaimer, I am not an FDA consultant. I am essentially just a few steps and perhaps a few years ahead of most people here today. We're currently at the FDA and CMS with an in vitro diagnostic device. Another project that's at Phase II SBIR, and have recently started a new project with an exceptional team where we will be submitting a series of Phase I SBIR grants and are in the process of designing our proposals by identifying our clinical indications first and working backwards to prove the feasibility in R&D, topic that we'll discuss later on. I won't be discussing the absolute specifics of every regulatory pathway here today, but my primary aim is just to introduce these concepts and, like you just said, just to help start thinking about your approach to these pathways for product development during the Phase I. So we'll start with the key functions and the organizational structure of the FDA regulatory centers and the relevant pathways. Go over relevant themes for medical device pathways with an emphasis on in vitro diagnostic devices, since that is what I have carried over to the FDA, and then touch on the general scheme for therapeutics and the recent emergency use authorization for COVID-19. The three key functions of the FDA mainly are the gatekeeper, the FDA's role as the gatekeeper to the marketplace for pharmaceuticals, medical devices, the nation's food supply, and cosmetics in the US makes the FDA one of the most consequential forces for human subjects protection in the world. So they are the enforcer, and they are responsible for the quality compliance of these items via inspection, will enact criminal charges on those entities who do not adhere. And they will do that through surveillance of pharmaceutical and product safety during development and post-approval with post-market monitoring systems, which we'll cover some later. So to start off, what are some of the common regulatory pitfalls for SBIR applicants? And it starts here with overall planning. It's always critical to create a project plan from the beginning with a well-defined go, no-go decision points. And that's important in SBIR grant writing as well. So in other words, plan with the end in mind and let that end result of labeling drive what you need to do. So the first key pitfall in that overall planning is to leave the research mindset behind and to transition over to more of a developmental mindset. Research mindsets can be problematic at times, and being able to change that internal mindset from research oriented to more of a developmentally oriented by asking yourself the question, how is performing this task going to help commercialize this product and get it onto the market? This is a common pitfall, and I've seen it firsthand many times with companies that I've been involved with, where interesting research questions sometimes take precedence over the end goal, and the end goal is lost in the mix. So having an idea of what is needed for regulatory success and accomplishing these tasks that contribute to the goal is important, not to waste money, to waste time, and to lose sight of your SBIR development, which leads into study design and making sure that you're integrating characteristics of every experiment with a specific clinical population in mind that may contribute to your regulatory intended use, which we'll cover some later. In other words, make sure that your targeted clinical population has a direct need for your product while keeping the feasibility of proving your labels or the claims to the FDA in mind. So one strategic approach that we were made bluntly aware of while at the FDA that we'll expand on later is to start small, a narrow indication, get approved, get to market, and later increase and expand on those clinical indications over time. So this is key to optimal product lifecycle management. And like I said, this is something that we learned of when we went to the FDA and we wanted the entire clinical indication to consist of all hypertensive patients. An enormous population, tens of millions of people just in the United States alone. So we thought that was a great idea. Well, the timeline of the study and the data that would be required by the FDA to prove our claims was simply not feasible. This is something not many small SBIRs can do, otherwise they wouldn't be in the SBIR program. And so it took time, but we reimagined our trial design, narrowed our indication, and ended up with a much cheaper overall clinical trial that could help get to market. Another example, if some of you have heard of the recent NephroCheck test that can detect AKI in post-heart failure patients and is indicated for that specific clinical population, may have and likely does have other indications that expand across all the AKI market, but narrow their population in order to get through the FDA, obtain CMS coverage policies, and get onto the market. And also something that can be lost if never been to the FDA before that is required is the design history file. So we were unprepared with our first product when we went to the FDA on design history file requirements and took months to catch up. So keep in mind the demand for documentation at the FDA is very high. So start these detailed reports early in the process and don't lose them or don't forget to really keep track of them over time. So also, it's always important to consider, I mentioned this before, the coverage and reimbursement policy. So when you're planning your regulatory approach, it's important to also try to cover your how you're going to pay for the test, and keep in mind who is going to pay for the test, because most physicians will not order a test or order a product or a therapeutic for a patient if that enormous overhead is going to fall on them, the patient. So eventually you must prove that your product not only benefits the patient and the physician, but also the healthcare system as a whole in order to receive that coverage and reimbursement policy. And the FDA has initiated a new pilot program, or reinitiated a new pilot program that is a FDA CMS review, and CMS being the Center for Medicaid and Medicare Services, where they can review your application at the same time and fast track that approval process and hopefully obtain that insurance reimbursement at the same time. So lastly, to keep in mind early in the process is GMP components. This is good manufacturing practice and making sure to audit your outside vendors aggressively. This includes CROs, clinical investigators, non-clinical testing facilities, contract manufacturers. And so we actually fell into a very significant pitfall at one point when we were working with a CRO who seemed very great, very intelligent, had everything lined up. We worked with him for 9 months, only to have our FDA consultant show up and audit him and inspect him. And unfortunately for us, he was not CGMP certified. It was a straight up lie, really. And in fact, the company could not be CGMP certified due to past infractions. So that was an unfortunate pitfall that we fell into. So it is important to audit these CMOs, CROs early, and to work with the FDA consultants very early in the process before you lose months on your timeline and a lot of money. So just a breakdown of the regulatory centers of the FDA. The centers we'll be focused on are the ones in blue. The Centers for Devices of Radiological Health, which cover medical devices and radiological products. The Center for Drug Evaluation and Research, which consists of most of the drugs and therapeutic biologics, such as monoclonal antibodies that we'll see later on. And we'll touch on the Center for Biologics Evaluation and Research, which are other biologics such as blood, cell, and gene therapy. The other regulatory centers are the Office of Foods and Veterinary Medicine and the Office of Food and Safety and Applied Nutrition. So we will go through these pathways specifically, leave out tobacco, and just keep in mind where you're going to be applying, where you're going to be sending your application to. So keep in mind that I'll refer to these later on as CDRH for medical devices, CDER and CDR, and I'll try to be mindful of reiterating what those mean and where those offices are when you get to that step. So just before we get started in the medical device regulatory pathway, just keep in mind the two main functional bases of the FDA are to show that the labels or the claims that you have on your device are both safe and effective and true to what you're saying. So there has to be a reasonable assurance that the benefits outweigh the risk of the device based on scientific evidence, and that the use of the device in the target population will provide clinically significant results. And that does not mean that by effectiveness or efficacy that you have to be 90% specific or 90% sensitive in a test or 100% accurate. You can go to the FDA and claim your effectiveness of straight 50% efficacy. And as long as that's true, and you show the clinical trial showing that's true, then you will get FDA approved. It does not mean you will get a reimbursement package, which is another topic for another time. Because if you don't show efficacious results, then it is very unlikely that an insurance company will pay you for your product. So to start, let's just start with the medical device regulatory pathways. We'll be going over ways that are products that are new to market, and products using substantial equivalence for a predicate device. And we'll focus mainly on the pre-market notification, or what is also called the traditional 510K. We'll touch on the pre-market approval, which is reserved for class 3 devices, and de novo, which is reserved for less risky devices. And we'll go over what all that means. And then we'll touch on what humanitarian use devices are as well. So just some common terminology that the FDA generally expects you to know if you are there, and what you would want to, and the language that you would want to use in your future SBIR grants are, first, most significantly, is the indication for use. And any FDA representative will drive this home because it essentially defines the specific circumstances in which a physician would order your test or device for a patient. So this includes describing your target population, what exactly will trigger the physician, whether it be a lab result, a test result, a comorbid condition, to place that order, and what is your device designed to do for those patients. For instance, diagnose, treat, screen, mitigate. There are devices that do many things for the patient. So making sure you discern and label that correctly, and making sure you use the correct language when you list your indication for use statement is very important. One common option, and one that we have used in the recent past, is an aid to diagnosis, which essentially claims that the results of the device will add a novel piece of clinical information to the already present standard of care, making that clinical picture more clear. And I mentioned NephroCheck earlier, and that is one way that they got into the market. Is they weren't trying to overtake serum creatinine in diagnosing AKI, but they wanted to aid in the diagnosis of the serum creatinine or estimated GFR result in order to make that diagnosis quicker. And so it is important to offer an advantage to that standard of care in making that diagnosis, and then showing that you're benefiting the patient and that the physician, and that will result in benefiting the healthcare system. That will come into play when obtaining insurance reimbursement policy. So by going this route in aided diagnosis, clinical trials are often more simple, as we have found, and don't often require extensive timelines of follow-up visits that are needed for screening devices, which we initially had wanted to go to the FDA with a screening device before we knew how extensive and how expensive running that trial would be, and how long that timeline was too. So adjusting and being able to pivot to get through the FDA is important. And then after a few years on the market, you should have the data to go back and be a screening device of sort, if that is what you wanted to do. So that's just one example of something that we went through. Another is it doesn't have to meet the high standards of being a confirmatory test. So consider this the indication for use early in the process when you're running your early experiments, and don't discredit a smaller subpopulation that may have a high market need and can be used to simultaneously streamline both FDA and CMS requirements without the high cost associated with a larger clinical population. So that is a strategy, and that is just important to be mindful of when you start collecting data. And this leads into the intended use statement, which is different and has different aspects based on whether you are an in vivo or an in vitro diagnostic device. And it is basically what will be said on the label when your device is on the market. So characteristics for an in vitro diagnostic, and these can be applied to other devices, and therapeutics have their own way of describing exactly what they will do. What are the physical parameters to be measured? So is your device measuring heart rate? Is an analyte from bodily fluid, in which matrix? So what is the specific target population? And knowing what that is will be true in all regulatory pathways in the FDA. And what is the clinical use? And being specific about what the clinical use is, such as a diagnose, treat, aid to diagnose, screen, and then finally, what is the clinical effect that the test result will have? What is the clinical outcome based on having that test result, or based on having that device, or based on having that therapeutic? So these are very specific measures of what is going to actually be on the label, and making sure that, and the FDA just wants to make sure that what you say on the label is exactly true in terms of both safety of what you're doing and effectiveness. So that is how the specifics of the intended use statement will work, which plays into defining a parameter device. So these parameters must be the same as a predicate device, which I mentioned earlier is the traditional 510K, which is the pre-market notification, which the FDA truly encourages everyone to go through that first. And there's no disadvantage through going through a traditional 510K first, even if there is no predicate device. But what they do say for what the requirements are for a predicate device is that the intended use must be the same, and the technological characteristics must be the same. So basically, the predicate device is a previously FDA approved, legally marketed product that was cleared through a regulatory pathway in the past, that is used as a point of comparison for your new device seeking to enter the market. And you have to do this by establishing substantial equivalence. And you can do this through either clinical or analytical studies. I think around 15% of 510Ks end up requiring clinical studies. But going down this route and establishing your diagnostic efficacy is the same as the predicate devices is an approach to getting through a 510K, which can be potentially hundreds of thousands of dollars cheaper than going through a different pathway, especially if you have a high-risk device, which we'll go through on the next slide. So this is just the thought process that will determine which approval pathway your medical device will have to take, and it determines what data, like I mentioned, the clinical and analytical studies that the FDA will require in their application. So there's a significant determination of cost and the timeline for review. So keeping these terminologies in mind is important, and to use these on the grants to show what the future of your product will be, and how the FDA works is important as well. So I mentioned before device classification and how that risk plays into which pathway your device is going to be shunted down. And there are 3 classifications of device, 1, 2, and 3. 1 being the least risky, low risk, and 95% of class 1 devices are exempt from regulatory review, and they do have to file for a exempt designation. So this classification, like I said, will largely influence where you're shunted. De novos are largely for lower class device, either class 1 that does have to go through a regulatory pathway, or class 2, which are moderate risk device. So most in vitro diagnostics, which is what I'm more familiar with, are largely class 2. If you have an in vitro diagnostic that diagnoses cancer, however, and the risk for a false result is going to be the initiation of toxic chemotherapeutic agents, then it is more likely that your device will be in the class 3. So as long as the device classification is based on risk, and the risk is based on, if for a device that gives test results, the potential outcome of receiving a false test result, whether that be a false positive or a false negative. And if you have questions regarding where your device falls, or want to make a case for a different class, determination can be made by submitting a 513G letter, and many companies do that. So if you do need to reclassify your device, so if you submit 510K, which is a pre-market, the traditional 510K, a pre-market notification, and they cannot determine a predicate device, or they rule that no substantial equivalence is made to a predicate device, then you will be shunted to a more costly pathway, which is the de novo or the pre-market approval, which are for technical new products. And with the de novo, you are automatically designated a class 3 device, and you have to be redesignated as a class 2. So if you do get shunted down that pathway, 513G letter is one way to prove ahead of time that you're not going to go down the class 3 pathway, and we'll show why you don't want to be a class 3 device here in the next couple slides, because of the high cost, timeline, and requirements even post-approval. So from phase 1 onwards, developers should have in mind how equivalence to a predicate would be established and what that predicate device is, theoretically. So if you're a device to diagnose kidney disease, your predicate device might be serum creatinine. And there are strategies to try to get that through the FDA. And a lot of times it won't happen, and you'll have to adhere to what NephroCheck did, which I mentioned earlier, and they went through the de novo process and were registered as a class 2 device. The FDA does have a search engine to help make this determination for you. And if you go through a pre-submission, which is basically just a pre-market, pre-review of your application, then they can tell you if your thought process and device classification is right. And the search engine that can help with that determination, help make that determination, is on the FDA website, labeled as a product code classification database. So like I mentioned, these are important considerations early on, and especially for when leading into your clinical indication and which pathway you're eventually going to go down. So just keep these in mind as we go through some of these pathways. This determination will essentially define the parameters of your indication for use and your intended use statement for the final pre-market review. So in essence, all developers must, the FDA does want all developers to take advantage of this free pre-submission program, which is that pre-market pre-review of your application, of your thought process, of your strategies. They'll review your predicate device, they'll review your clinical trial protocol, they'll tell you whether or not they agree or disagree with your device class. And and going on to the next slide, it is all driven by how you word your intended use statement, how you label your device, and what claims you're making. So the pre-submission program, this is also equivalent to the pre-IND, which is for therapeutics, which we'll go over a little bit later, and the pre-NDA also for therapeutics, which we'll go over later, where the FDA will critique every characteristic of your strategy. And some important items, like I mentioned, they'll comment on, and like I mentioned, they will tell you whether or not they agree or disagree. So you can put questions in here, but in general, they are not going to give you a better strategy than what you have already put on your application. They're not going to tell you, oh, if you change it a little bit this way, the clinical trial will be much less expensive and it can be streamlined. They'll just tell you if they approve of the strategy that you put down and whether or not that would eventually lead to, assuming positive results, a pre-market approval. So for instance, one recent pre-submission meeting I was a part of for an in vitro diagnostic led us to eventually entirely change our clinical protocol, our trial endpoints, re-specify and narrow our targeted population, and we ended up with a much less expensive clinical device. So that was one instance where the FDA had a lot to disagree with on our pre-submission. So the next one went much better. So that, like I mentioned, this is a free program, the pre-submission meeting request, and no money is wasted, and you could end up with a much less costly validation study. And they do recommend you do this before initiating any kind of clinical trial because of the high costs associated with clinical trials. And this also goes for therapeutics as well. So keeping those items in mind, this is the first pathway, like I mentioned before. This is for new devices, or this is for the traditional 510K. This is where you're trying to establish a predicate device. It's what you'll go to first, and you'll see the low cost here, especially with small businesses, is only several $1,000s. So there's not a lot of cost expenses associated with submitting this application. So this will include all class device, and your strategy for establishing that predicate is going to be important on whether or not you can actually achieve approval through this pathway. If the FDA does review your application and says, you could not establish a substantial equivalence given the evidence that you have shown us to this predicate device, then they'll go ahead and they'll shunt your application to as a designated class 3 until it's reclassified as class 2 to either the pre-market approval or the de novo pathways. The pre-market approval pathway, as you can see here, the application costs are upwards of several $100,000 for a small business, closer to $100,000. And on top of that, you have to submit annual pre-market approval reports after approval, which is another several $1,000s. And with larger businesses, there's also an annual registration fee on top of that. So it's a very costly process, and the price increases every year by $1,000 to $2,000. So until you get to by the time that many SBIR ones are to the FDA, it could be around $100,000. And this is also the longest timeline for review. So this timeline, 180 days, really is just from the moment that you submit your application to when they are supposed to give you back an answer. And as we have found out, sometimes they're not, they do not meet that deadline. So this is a rough timeline. So hopefully they adhere to that timeline, but oftentimes it's, at least in one of our cases, it was it was even longer. So this is the most stringent, timely, and costly regulatory category for medical devices. But if you are able to reclassify your device as a lower risk, class 1 or 2, that's new to market, then this is a much less costly option, around $25,000 for small businesses, and lesser timeline for approval. And the data that they require in de novo is also much less. There's no annual post-market reports that have to be given. So once you're on the market, you're on the market. So de novo, and so being reclassified as a class 2 would be very helpful, especially if you are a small business and money is a concern. Lastly, I just wanted to touch on another strategy called the Humanitarian Device Exemption. So first, you would have to go to the Office of Orphan Product Development and be classified as a humanitarian, get the designation as a humanitarian use device. This is defined as affecting an estimated population of less than 8,000 patients. So not a huge clinical population, and so not a huge market. But if you do see a potential within your disease, within your non-rare disease, a subpopulation, this is defined as an orphan subset. And you can use that orphan subset as a target population to basically get streamlined through the FDA without having to show near as much data for the larger population. At least you can get on the market, generate sales, and then come back with a larger clinical population or larger target population after it's already been on the market and proven its safety and effectiveness. So this is a route that some people take as well. And some advantages here is you are exempt from the effectiveness requirement. And so they really just look at safety, and there's no fees involved, and the timeline for approval is 30 days. So it could be tough to do if you're looking at an orphan subset, but if you do have a smaller population, or do think that there is an orphan subset within your non-rare disease, then this could be another potential option to really make it through the FDA faster than some of the other pathways, and much cheaper too. So just wanted to touch on the EUA process briefly. It's the Emergency Use Authorization. One has been issued for COVID-19. This was done by the Health and Human Services on February 4th of this year. So this essentially provides special features for certain pathways that will require significantly less stringent evidence for approval. That will ultimately accelerate and expedite products and therapeutics that either diagnose or treat COVID-19 or a related pathology due to COVID-19. So COVID-19 related heart failure, COVID-19 related kidney disease. So there is a long list of items in which your indication can target for EUA pathways. EUAs in recent past, for example, have also been issued for the 2016 Zika virus, the 2014 Ebola, the 2013 H7N9 flu. So basically this is done by the Health and Human Services, but it is important to note here that once they revoke that public health emergency, the EUA is revoked by the FDA. And that product that is currently on the market would also be revoked from the market. So it is encouraged that that you also go down the pre-market review pathway at the same time, or at least sometime in the near future once being on the market. So that way your product is not pulled from the market and sales just immediately cease when that EUA stops. So that's just something to keep in mind for the EUA, and we'll touch on some of those pathways a little bit later on. And so just to round up the overview of medical devices, this is just an illustration. It's a good illustration for visualizing everything that we just went over. And so you can apply this to the offices that you would also apply to as well, that we went over earlier too. So, and you can see how the device class here, and I think it's important to see how the device class will define the pathway that you're eventually going to go down with your products, and the information required for each path. And you can see down here the general controls, special controls, clinical data become intensely more extensive with each class that you progress down. And you can also see here that the 510K establishing substantial equivalence to a predicate device is with first step, and this is what you would want to go down first given the significantly less cost, and if it's not established, then you're either sent to the de novo or the pre-market approval pathway. So this is just a good overview of everything we just talked about and getting everything visually oriented in your mind. And so now we'll just go ahead and touch on some regulatory pathways for therapeutics. We'll go over the general scheme. They have several different categories. If you're going to be prescription, generic, over-the-counter, I'll touch on a couple special populations, especially the EUA for COVID-19. But for the most part, I'll focus on the prescription use, investigational new drug, different categories, different types of investigational new drug applications you can submit, and being sure to submit a pre-IND before actually going down that pathway after animal models are complete is also very important to do, just like the pre-submission is for devices. So they're equivalent in the process, and you don't have to do it if you are familiar with the regulatory steps involved, but it is highly encouraged and is free, and it's not a lot of time wasted. They usually turn around a pre-submission in a few months. And then we'll go over kind of the new drug application, how that differs from the biologics licensing application, which are basically just synonymous of each other based on what your therapeutic is classified as. And before getting into it, just to start with a visual here, since this does have kind of a complicated nature, it's important to look at the process and your target identification in animal studies before submitting the IND application. Then you would go through phase 1, 2, and 3 trials, and then submit your pre-NDA, and then later your NDA. And this is synonymous with the NDA, like I said, a BLA, and I'll show a visual for that later as well, before moving on to the final phase of your process. Investigational new drugs, there are 2 categories, commercial and research. I believe many people here are commercial, and that is where data is collected for clinical safety and effectiveness for a new drug application, an NDA. A research investigational new drug in human studies is just to advance scientific knowledge. And so you can also submit one for a research IND as well. There are 3 types, investigator, emergency, and treatment. Most people here will be investigators, so it's a study to collect data for an unapproved drug or an already approved drug for a new population. An emergency just allows an experimental drug to be used in an emergency situation. You don't have to go down the pre-IND, IND pathway or review process. It's much faster. If you do get an emergency IND, then there is a very accelerated, expedient process that that you can go down. And then there's a treatment IND. So that it differs from an emergency, because if you are at the very end phase of your clinical trials, human clinical trials, and showing strong promise, then they can go ahead and submit that to treat patients that have a serious or life-threatening condition. So this is the investigational new drug pathway. This is how it breaks down after you submit your pre-IND, and you're going for your IND to translate from animal studies into human studies, which you must have the IND before you start any human studies for therapeutic. And it's basically requesting the FDA for permission to initiate human trials. And this is submitted after pharmacology activity and potential toxicities are observed and recorded in animal models. And so once you submit your IND either to the CDER or the CBER, whether it be a drug or a biologic, and we'll go over what the differences are there. I'm not sure anyone here is in the biologics versus drugs. But once submitted, they'll submit to 4 different centers of statistical analysis from medicinal, chemistry, pharmacological, and statistical point of view. They'll review the safety of that drug, not the effectiveness, but that will review the safety and tell you whether or not you can proceed. And if the review is complete, then they will issue your IND and you can initiate your phase 1 clinical studies. Just a little bit about drugs versus biologics is that the main point here is that a monoclonal antibody, even though generally thought of as a biologic, would be submitted to the Center for Drug Evaluation and Review, not the biologics. Biologics, the CBER, includes other products such as cytokines, growth factors, proteins have been extracted from animals or microorganisms, non-vaccine immunomodulators, immunotherapies, thrombolytics, stuff like that. So the monoclonal antibody therapeutics are a CDER product, and that's just one of the differences to keep in mind. And like I mentioned before, once you have gone through that IND process, you have started your phase 1, 2, and 3 clinical trials. Phase 3 clinical trials are showing that you are supporting your therapeutic indication and that you don't have the toxicities that are that would rule out your drug. This is just a process of how you will then submit. You'd want to submit a pre-NDA to have your therapeutic reviewed, your clinical trial information, and then you would submit your new drug application or a BLA at this point. And this is just an illustration to show that the next step in the process and what would follow after. So these are, and I do mention that another difference between drugs and biologics is biologics can cost a lot of money to get onto the market, sometimes upwards of hundreds of millions of dollars. So if you are in the therapeutic business, I know that some people, and I'll go back to, if you are in the therapeutic business, even drugs themselves can cost upwards of tens of millions of dollars easily once you get to this IND human study phase. So many SBIR applicants like to get out and sell in this animal model study when they're submitting their pre-IND, or once they have gotten their IND, or once they've started human trials, because once you get to this point, the supporting these trials are extremely expensive, and there is a very significant failure rate on top of that. So selling out to a pharmaceutical company that may be interested and can support that without putting them out of business is a prominent strategy to not bankrupt your program. And then I just lastly want to touch on the EUA for COVID-19 therapeutics and some of the expedited development programs. So what I really wanted to get across here briefly is that there are many fast track or accelerated or expedited programs that you can go through now that an EUA has been issued for COVID-19. So the expedited development programs are fast track designation, breakthrough therapy, accelerated approval pathway, and expanded access versus, and you can also apply for an expanded access versus an actual clinical trial. And this is, then you'll be granted a EIND, and it's important here because of the significant differences in what is required in order to get market approval. So clinical trials, you have to include as many people to show statistical significance, whatever that means to your product. And the FDA doesn't really help you with that, determining how many people you need. You really have to just show a strong statistical significance and that your claims are true. Expanded access don't adhere to those strong statistical findings that you're required to have. And in some cases, you can have as few as just a few patients, and some very emergent cases as low as 1 before it's approved. And I am not aware of what's required in order to get down the expanded access pathways, but I do know that if you are interested in therapeutics for EUA, then these fast tracks, these expedited programs are something to into. But what is important to know is that you do also want to go through the pre-market IND review simultaneously, because if you do not, once that EUA is revoked, that therapeutic will also be revoked from the market. So just keep in mind that submitting, they do have a pre-IND consultation program, and this has replaced the pre-EUA program. The FDA switched this over several months ago. They have done away with the pre-EUA program for COVID-19 because of the lack of information that was needed in the pre-EUAs in order to get the drug fast-track approved. So the pre-IND is more inclusive, and the FDA felt to get more drugs approved faster. So don't go down the pre-EUA program in this case, and you'll find that when you start looking into it. And they do, if you do have any questions or concerns for this EUA for COVID-19, you can go to the free, the FDA now sponsors a free pre-IND consultation program. So they can answer any questions for you regarding this path or regarding any of these pathways. And just to touch on some of the other pathways, I just want to know, want everyone to know that generic drugs have their own pathway, an abbreviated new drug application. Over the counters, you can submit a monograph, which is a much more simple version of an ANDA or an NDA. You can go down over the counter through an NDA or ANDA, but an OTC monograph is generally used. There are biosimilars, and just if you have an orphan drug, or if you do have a population, then the Office of Orphan Drugs that I mentioned before, you can submit an application down that pathway to get an orphan drug designation for a disease that affects less than 200,000 people. So just to wrap up, I know I'm a little bit over, I just wanted to touch on how all this fits in for SBIR applicants and what to think about. And hopefully those thought processes that I talked about in the beginning and some of those common pitfalls, you acknowledge throughout the presentation and kind of apply to your own product or your own therapeutic. So hopefully you now have somewhat of an understanding of what that regulatory mechanism your product will be, if not just at least a visualization of how that regulatory process occurs, and just making sure that you're acknowledging the pitfalls in your SBIR grant and showing that you've designed a plan to overcome those pitfalls. I think is a very strong way, and I think a lot of grant reviewers, I think there's people on my team that serve as chairs of many SBIR study sections, very many study sections, and there has been a new wave of, even in SBIR, people focusing on a strong, at least 3 to 4 sentences of their plan for an FDA pathway. So it's not that you have to have everything worked out in your phase 1. These are just considerations to think about. And so that way you can work around and develop as you start to generate data, and as you begin to pinpoint certain indications or certain clinical populations that you could see your device being helpful for, that there's a market need for, that would not only get through FDA quicker or faster than a large clinical population, but also obtain an insurance and reimbursement package from CMS, because ultimately approval is not the most important part here, even though it is required. Obtaining a CMS is a Center for Medicaid and Medicare Service insurance reimbursement package is also necessary as well. So do consider the FDA CMS parallel pathway, and that's just where CMS will sit in on your on the FDA's review of your product. Hopefully expedite that process. So this is the last slide, and I hope that I was able to help with some people's thinking of the regulatory pathways, and if there's any questions, then I can try to answer them as best I can.
Thank you so much, Aaron. Please put your questions in chat. We'll go through those. There was a question on whether the presentation slides will be shared with meeting participants, and the answer is yes. Both the slides and the webinar recording will be posted on the Accelerator Network's website. That's where in the section of SBIR webinars and presentations in the next few days, as soon as their video is processed. But yes, both the slides and the recording will be available. Any more questions to Aaron? I think that this has been very useful presentation. I think that it really addressed the most important part for that particular population that we're hoping to help right now. People who are either in their SBIR process right now, applying for it. We have several people here on this webinar that are about to submit their application. So that was very helpful for their submission, and others that are thinking about SBIR applications. Any more questions? And then I see that Jackie just posted their website for Accelerator Network. Thank you. Okay, so the question is from Carlos from Puerto Rico. Can you comment how broad the indication should be on an IND for a therapeutic, phase 1 versus 2 and 3?
I'm not sure on therapeutics on broad versus narrow. I know that in general the same themes apply, but therapeutic indications I wouldn't want to comment on and lead you down the wrong way. But there are, if you do, there is a way to figure out, and there are FDA guidance documents. If you visit the FDA website, they have an IND guidance document. They have several IND guidance documents that are total up to probably a couple hundred pages long, but I know that the answer is likely in there for a better strategy, and it might help you find a better strategy once you get there.
While we're waiting for people to type, the questions, I have a question. Could you comment a little bit on selection of FDA consultant, when you will need, that? At what point do you need to bring a professional in, and how you select the right one?
So yeah, I did mention early in the presentation that it is important to bring them in early to at least have them on hand. I think that just based on things that my team has said, it's never a bad idea to include a letter from your FDA consultant on even a phase 1 grant, even though they won't play much role during your phase 1 applications. But when you start to develop your product and you're starting to get those materials from your CROs, it is important to audit them early, because like I said, we had a 9-month waste of time for one characteristic of our product, because once we actually did audit them, we figured out that they were intentionally lying about their CGMP certification. So I don't know how common this is, but it did happen to us. So bringing them early and just auditing them early before you waste a lot of time and money, I think is probably a good idea.
And what is the typical cost of bringing an FDA consultant? Are they charging by the hour, or you can negotiate for a specific task until completion?
Our FDA consultant was grandfathered in from a previous company, so he's a very established representative, and he's close to $400 an hour. But you could find cheaper ones for lower, for 300, but perhaps less, or maybe a less experienced representative. Okay. But yeah, that's how we pay him. So we don't contact him unless we really need him.
Okay, so essentially, that consultant is on call whenever you need it, and then you pay by the hour.
Yeah, you got $400, and you got him for an hour.
Okay, yeah, that's good business.
Yeah.
Could you also, I had a question in advance of that webinar. I'm not sure that the person was able to join. You talked a little bit about EUAs, and the question for some is, okay, so this is the route we're taking now. We get it. What do you need to do while you are operating under EUA, knowing that hopefully at some point the COVID situation will go away and you will need the real approval? At what point and how you process while operating with EUA?
Well, the FDA recommends as soon as you have the data to go ahead, the animal model studies done and complete, to go ahead and start that that pre-IND, that IND pre-market review pathway at the same time, because if the public health emergency ends in a week, then your product is immediately revoked, so from the market. So if starting that process now, because it is a long, costly process, what is an advantage is that you will be generating hopefully a lot of sales from being on the market through the EUA. And some of the features that I've come across in some of these pathways do allow for an extremely accelerated pathway approval for the EUA. So you could get on the market extremely fast and for extremely low cost compared to the normal pathway, generate sales, and then use those sales to put into your normal pre-market review. And that's just what most FDA consultants, and that's what most FDA people would advise that therapeutic to do. And in addition to that, you're already generating data from human studies. So you can use those, the results from the outcomes from the patient population that you're currently using on the market to be part of your pre-market review package.
Okay. One more question that I was asked also in advance. Any difference in terms of your FDA strategy for SBIR companies versus STTR when you have a university involved? Any other role that the university needs to play in all this, or it's still the same, the company needs to lead it?
As far as regulatory, the regulatory pathways are concerned, having a university involved wouldn't make a difference. It's just about proving your product is safe and effective for your clinical population. As far as I'm aware, I have been a part of STTR once in the past, and I don't think that there's any difference once you get to that point.
Okay. Well, thank you so much. Any more questions from listeners? Hey, going, gone. Well, thank you so much, Aaron, for a great presentation. Very, very useful. Already received several emails from people who were on that webinar saying that it was very, very useful. I think it was particularly useful to hear those things not from someone who is selling their services as an FDA consultant, but someone who is a near peer. I think that it just makes it much more relatable. Thank you so much for doing that. I presume it's okay if people will ask questions later, that they would be able to, that you're okay with us sharing your contact with them if they want to talk maybe about collaboration or anything like that.
Yeah, absolutely. And thanks for having me. And I hope it was helpful for at least some people, and hopefully you're just able to avoid some of the missteps that we went through as we were going through these processes too. Because it is complicated and it's very tough to try to grasp an understanding of what regulation, what these FDA regulatory requirements are. So I do completely understand.
Thanks again, and there are several messages in chat thanking you for your time and insight. Great info, great presentation. Thank you so much.
All right, thank you. Thanks for having me. Appreciate it.
Thank you. Bye-bye, everybody.
All right.